here we go. So you should have finished up the cell size lab activity here the other day, and so now we're going to just review that for a little bit, and then see if we can't tie a few things together, and then get you working on the phases of mitosis. Going to have you do a few drawings and insert your drawings into your interactive notebook. So remember the reason that the reason we even talk about cell cycle and cell division is largely due to the fact that our cells can only grow so big, right? And then they need to divide. And there are two major reasons for that. One of them, and I like to give the analogy of, think of the nucleus of a cell as being like the media center at Eastridge. When Eastridge first opened, there were only 1,000 students there or so because we didn't have a senior class and some of the other classes were a little smaller. So roughly half the number of students were there as are there now. So you can imagine 1,000 students going to the media center to check out books for class. Fairly easy. Not a lot of pressure on the media center to get those books out. Okay, so if the nucleus is like the media center, right, providing information, right, and the students are like the cell and the parts of the cell, right, ribosomes and mitochondria and whatever, looking for that information, they had fairly easy access to it. But as Eastridge grew, right, the number of students is now nearer to 2,000, the media center is still the same size. And you can imagine the pressure that that puts upon the media center now that the number of students coming to it is so much larger. It's kind of the same idea with a cell. As the cell gains more ribosomes and more um, parts, lysosomes, mitochondria, whatever, all those things are requiring something of the nucleus, but the nucleus is only so big, right? The nucleus doesn't grow, kind of like the media center hasn't grown at Eastridge. And so the cell has to divide. Another major reason number two, that cells need to divide before they get too big had to do with the lab activity that you did, and that's the cell size thing. And hopefully you got to see with the math that as surface area grows, of course, so does volume. But they don't grow at the same rate. And if you think about that mathematically, it makes sense. When you're talking about surface area, you're talking about squared area, right? Centimeters squared. When you're talking about volume, you're talking about cubed, right? Centimeters cubed. And so the volume is growing at a faster rate than the surface area. And if the surface area of a cell, of course, is the cell membrane, that cell needs to be able to get materials through that cell membrane to the inside of the cell. But if the volume increases too much, the cell can't do that. So two major reasons the cell needs to divide, two major reasons: to give the nucleus the ability to still do its job without getting overwhelmed, and surface area to volume ratios. So just a quick review of that. Okay, so we're mostly going to be talking about eukaryotic cell division, but of course prokaryotes also divide. They also have genetic material, just nowhere near as much, and it's not organized the same way. Speaking of organization, right, remember, it isn't just DNA that has to sort of be divided and separated out during cell division. There are proteins that are involved. Here's sort of a look, right? If you were to blow up what a piece of a chromosome looks like, very condensed, very efficient storage. Because remember, every cell has about 10 feet of DNA in it. Every one of your cells, except your red blood cells, has a lot of DNA in it. And you can imagine the process required to duplicate all that. Imagine walking into the media center and having to make a copy of every page in the media center. it'd take you a long time for all the books that are in there. And so uh, if you have an efficient process to do that, you're that much better off. Okay, so prokaryotes divide, binary fission, um, similar idea, right? Essentially, the genetic material doubles first, then it is split into the two uh, sides of a cell, and then that cell ultimately separates, and now you have identical genetic material, at least in both cells. Of course, the rest of the cell material got divided out between the two new cells. Okay, eukaryotic cell division is the main one we're talking about because these are types of cells. There are three major phases you want to think about in the cell cycle. Don't mistake the cell cycle for cell division, right? The cell cycle includes sort of the normal life of a cell too. Cell division is really just the reproduction of the cell. So 90% of a cell's life is spent in normal cell life, called interphase. So if you're a liver cell, for 90% of your life, you're just doing liver cell stuff. But for about 10% of your life, you have to go through cell division, right, for those reasons mentioned already. So to get into that in just a little more detail, there are three parts to interphase or normal cell life. The majority of the time, that cell is spending its time in G1. It's just doing what it's supposed to do. But it also is growing, right? It's adding proteins, it's adding new organelles, right? But not new genetic material, right? So there's that demand put upon the DNA from these new organelles and new proteins. Then a cell goes into the S phase, and this is the synthesis phase. So now it's starting to synthesize and make that new DNA because it's recognized, uh-oh, getting too big, we need to divide. So it starts doing that toward the end of its normal cell life. Okay, the S phase, synthesis. G2 is a growth 2 phase, so G means growth, S synthesis. So in the growth 2 phase, now the cell is making specific parts to prepare for division. Okay, so spindle fibers, that type of thing, start to form. What does it look like sort of globally, the bigger picture? During the S phase, the genetic material represented here in A is duplicated, right? So literally a mirror image, a copy is made. Now that that genetic material is made, the cell can go into mitosis. And this is where the nucleus divides into two nuclei ultimately. And what do you get out of this? You get a new cell with the same number of chromosomes as the original cell and a new cell with the same kind of chromosomes. So in other words, it's identical, right? It's genetically identical. So when you're creating or copying over and making genetically identical material, that's a form of asexual reproduction. Of course, we'll talk about sexual reproduction later, and then you're not getting exact genetic copies. You're getting a mix of genetic material. So there's four phases to the dividing of this genetic material that happened during the S phase. So the S phase, the material is doubled. Now we have to go through the phases to divide that material out. Okay, and this is a little bit of a false reduction, so to speak, because ultimately you could talk about more phases, and as a cell's living, it's just doing these things, and we just kind of take snapshots and talk about these phases. So we'd like you to draw this in the notes that are provided. You should be able to draw these. And phase number 1 is prophase. Okay, and the way to remember this one or think about this one is things are sort of breaking into pieces. Nuclear embryo breaks down, etc. So you can see the main thing we're focusing on here is the duplicated genetic material is starting to prepare to divide. The second phase is metaphase. Metaphase, we think middle. And why? Because these chromosomes that are about to divide align themselves in the middle of the cell. So if you're viewing this under a microscope, right, that's what you would see. Okay. Okay, the third phase is anaphase, and this we always think of away. So take a look what's happening. The genetic material is now moving away from the middle, right? And you should, if everything goes well, everything disconnects properly, have identical copies of genetic material going either direction in the cell to either side. And then the final phase of mitosis, right? This doesn't take very long. It isn't a big part of a cell's life. Mitosis is telophase. And a nice way to remember this one is to think 2. This is where the genetic material is fully polarized, right, to the two opposite sides of the cell. The nuclear envelope is starting to reform, and genetic material is starting to sort of uncoil again. And then finally, cytokinesis, which is the second part of cell division. So cell division, right, first of all, there's a cell cycle, the big umbrella life of the cell. Then there's cell division, right, and that includes mitosis and this part called cytokinesis. So mitosis is the splitting of the genetic material. Cytokinesis, as the word implies, is the splitting of the cellular material. So now the cytoplasm moves and splits into the two separate cells. If everything goes well, you have genetically identical cells. Okay, so again, identical to the original cell. This allows organisms to grow, right? Because we don't grow by getting bigger cells, we grow by adding cells. It allows us to replace damaged tissue. And some organisms use this strictly as their means to reproduce, right? They're just reproducing themselves asexually, making clones of themselves. Okay, cytokinesis is slightly different in plants versus animals. Not a huge deal, but remember, because plant cells have a cell wall, there also has to be the consideration that a cell plate forms before a new cell wall forms. Otherwise, the rest of it is essentially the same in eukaryotic cells. So there you go, sort of the whole cell cycle, right? This isn't represented very well, much of the cell's life is spent here. 90% here is interphase, right? And during interphase, you have G1, S, and G2 getting prepared to go through cell division. So this whole big part here is cell division. Even though this is a fraction of a cell's overall lifetime, it's represented here. You can see this is an actual cell being looked at under a microscope in prophase, metaphase lined up in the middle, anaphase moving away, telophase at the two opposite ends of the cell, and then cytokinesis where the cell has actually divided its genetic material, right? So mitosis and cytokinesis, the two parts of cell division. Okay, oops, I know that was a lot, but you should be able to hopefully just pause and draw the pictures as you need to for the notes. And take care. We'll see you next time.